Hello. My name is Martin. I'm going to talk about mobile deep brain recordings doing real-world and imagined navigation. I do not have any disclosures, so let's start. I structured my talk in two parts. In the first part, I will talk about mobile deep brain recordings using invasive methods to study the deep brain while people are moving freely in space. And in the second part, I'm going to discuss how close we can get with non-invasive technology, meaning scalp-EEG and computational methods, to reconstruct similar signals than we actually record using the intracranial ones. So let's start right away with the first part. We have a pretty unique lab setup where we have the opportunity to record directly from the medial temporal lobe areas, or in general deep brain areas, using invasive technology. That is why the reason for that is we work with very special participants having chronic implants in their brains for the treatment of epilepsy. So they have these responsive neurostimulation devices implanted chronically. So if you would meet one of our participants, you would not realize that they have actually brain implants. So they come into the lab and allow us to disable their stimulation just for a few hours, so we can record from their brains. So we use the recording capabilities of these implanted chronic devices to access their brain activities invasively. And because it's chronically implanted in their brains and under their skull, we actually can record from particular brain regions that we select a priori, so invite only these participants that have particular localizations to study their brains in our lab setting or in the real world while they move around. Scientifically, we're interested in the relation between spatial navigation and episodic memory. As you can imagine, personally, I still get lost sometimes in the mall downstairs, but how we can solve this task is to remember which turns we took, when and where, and that might be conceptually very similar to what events happens in our lives, at what times, at which places. And based on these relationships, looking at the neural underpinnings of these behaviors, previous studies in animals showed very well structured neuronal sequences that appear in an orderly manner. Not only when the rats are running in these mazes, but even when the rats are coming back to this wheel and run in this running wheel, even though they just run in this running wheel and do not really navigate in space, these authors were observing these neuronal sequences that fire in a very particular order or sequence. And to establish these sequences, theta oscillations are instrumental. So if you would block theta oscillations in the hippocampus in those animals, these sequences would be disrupted and also behavioral performance would be lower. However, in humans, these theta oscillations are not continuous as in animals, but they appear in short-lasting bouts, lasting for about 700 milliseconds to 1 second, which poses the question, what is actually the functional significance of theta oscillations in the human brain? And to answer this question, we designed a task where we did admittedly something very similar with humans. So we asked them to navigate in space, in our lab space, and you already can see here how large our tracking space is. And these people have, as I mentioned, these sealed chronic implants, which allows us to study their brains while they actually move in space in our lab. So first of all, we just show them this illustration of the maze we would like to study, just on a computer screen, and they had to remember where to walk in the room without anything in the room indicating where they actually need to walk along. So then they start and try, they come back, and we show them their motion tracking trajectories on a screen overlaid with these ideal paths, which leads to themselves learning walking on these trajectories very efficiently. On the right, I show a time-lapse illustration of the same person doing a rightward trajectory walk. On the lower left, I show the implantation sites indicated in red, where we had the electrodes in this particular patient. And on the lower right side, I show medial temporal lobe recordings while this participant did this task, and again we look at theta oscillations while they actually walk in space. Looking at the results, on the top left I show the behavioral results showing motion tracking trajectories from the top of all 5 participants that were coming in for the study. As you can see, all of the participants learned to navigate these trajectories very efficiently. And on the right side, I show time frequency activities for the leftward, which is this one, and the rightward walks on the top and the bottom. And you already can see very salient time frequency activities in the theta range, aligning up at particular moments in time. And those moments in time are actually when they're about to do a turn. So we plot this activity in the 4 till 10 hertz range, that is the theta range, and then we can combine neural activity and motion trajectories to plot it on the same figure, which I show here on the lower left. So when you look at this plot, you already intuitively can see how the activity is related to the motion trajectories. So anytime a participant is approaching a turn, theta power is going up, going down after the turn, and goes up at the next turn again. And this is a pattern we see over and over again. On the lower middle, I show activity aligned to these turns to investigate the precise temporal structure of this phenomenon. So you already can see 1 second before they actually physically turn, you see activities in their brains related to these upcoming turns. And on the lower right, I show theta increases along with behavioral data. So in order to do a turn, they slightly slow down, which I show here in the velocity curve. But even before the first significant decrease of velocity, theta is already at its maximum, suggesting that this is not a trivial speed effect. And here I show angular velocity indicating the physical turns, and again, these brain signals lead the behavioral signals here. And we showed this effect in all the participants we recorded so far. So these are the results in the left anterior hippocampus in all of the 5 participants. Participant 2 did an additional turn for who knows why, but I said it's okay, you can just do what you're about to do as long as you do it consistently. And you already can see also in this participant, even though this participant did an additional turn, the effect is still there. And here I show a video of a participant doing this task. So on the top left, you see the real-world camera. Here the motion tracking trajectory from top, a bar plot illustrating theta power, and you already can see here the raw EEG data while the participant is doing this task. So I just play this and you see quite intuitively what happens. Theta oscillations getting stronger. And again, even if the participant is turning in the middle of the room, signals are getting stronger and more oscillatory, and that is also true for right turns, which I show here. And also in this last example, I hope to convince you that when we look at the signals, it's quite intuitive what happens. Signals are getting much stronger and much more oscillatory before they actually perform a turn. But what about imagined navigation? I talked a lot about real-world navigation. What I did not tell you so far is that when they do this real-world navigation task, they come back here onto this treadmill, and in one condition they just walk on the treadmill without any additional task, and then there is a second condition where they walk on the treadmill while imagine themselves walking in the real world. So we have the 3 conditions: real-world walking, a control condition where you just walk on the treadmill without an additional task, and the third one when they imagine themselves walking on the treadmill. To investigate imagined navigation that is only in their minds, we used a metric where we split the data in 2 parts. Here I'm showing single trials illustrating theta power in colors, and you already can see in the single trials whenever these theta bouts illustrated in yellow are happening, and they're also well aligned in time. So then when you split the data half, what we did, we averaged in each pool A and B and correlated the resulting mean signals. So if these mean signals are highly correlated, that means we have some kind of temporal structure that is meaningful and task-related, and if not, it might be no task-related activity at all. using this metric, and we did this like 1,000 times to get a robust estimate. If you do that and you look at these three conditions I just mentioned before, we see high temporal consistency or correlation in the real-world walking data, nothing at all in the control condition that is treadmill walking without imagination, and as soon participants start imagining themselves walking on these trajectories as they learned before, we see something in between. So we see significant temporal structure that is significantly higher than in the control condition in the imagined navigation condition, which was quite striking to us. And each of these small circles is one channel pooled across all participants. So in total we had 18 channels in the medial temporal lobe area. And here we show the relation between these channels, between both conditions, so the higher the temporal consistency in the real-world walks, the higher it is also for the imagined navigation condition. And you already can see here this nice linear correlation. And I wanted to highlight that these 5 red circles are actually one channel in the anterior hippocampus for each participant. So each participant showed that a cluster, an anatomical cluster in the anterior left hippocampus in each participant showing the highest temporal consistency. And if you take this data and align it accordingly using the start and end time of their imaginations and plot this theta dynamics on the trajectories, the real-world trajectories, you already can see clear parallels between the real-world and imagined navigation data and how nicely it lines up. And actually also these two theta dynamics curves on average are significantly correlated. Despite the fact that during imaginations they don't actually physically turn, there is no visual flow and there are no environmental constraints they can work with. They just imagine themselves walking on these trajectories which they learned before. But what about, so we were very amazed by these results. So we thought about, can we actually replicate these results only using non-invasive methods? Because textbook tells us that we actually cannot observe activities from deep structures using scalp EEG. So in the same participants, using the same paradigm, we simultaneously recorded scalp EEG and tried to source model activities in deep regions, including the hippocampus. So we started to record scalp EEG in this mobile setup and took a 3D scan which indicated which electrodes or where the electrodes are actually. So here we just did a labeling of the electrodes. So we have a very precise model where the electrodes are on the scalp, and that we co-registered with a model we derived from their T1 MRI scan, where we model not only the scalp surface but also where the brain is localized in their head. And using a brain parcellation or an atlas, we already then can co-register this activity with particular structures such as the hippocampus shown here in yellow, which gives us the whole geometry of the setup. So we know exactly which brain structure is where relative to the electrodes. And this model we can use to set up a forward model, a mathematical model that describes, assuming you have a particular patch in your brain being active, where would this activity spread to the scalp. And that is what we do for all the sources in the brain, covering the whole gray matter volume. Having this forward model set up, we want to reverse or inverse this relationship to estimate the sources underlying scalp activities using inverse models and source modeling. If you do that, you end up with a blurry reconstruction of the actual ground truth activities. However, the inverse model is not uniquely solvable, so we need to assume additional constraints. One popular approach is to just model a few dipoles and then compute the localization and strength of these dipoles. However, we use the different methods that is very popular and established too, using distributed source modeling techniques, which assumes smoothness, meaning similarity between neighbors of neighboring sources. In our case, we used Loretta for this inverse model. And to answer the question if that actually works, we focused on our work on previous work of our group, where we used non-invasive scalp EEG. So you can see here two streams of processing. The top one is the non-invasive processing stream, where we use these EEG source modeling and source reconstruction techniques to reconstruct signals covering the whole gray matter volume. So it could be anywhere in the brain. We do that for every point is, you can see these small points here covering the whole brain. Non-invasively, only using the non-invasive data, and then we compare the source dynamics with the ground truth activities that we actually recorded, in this case back then in the thalamus, as indicated by the implantation site here in this small blue dot. And if you do these computations, you actually can see that the highest correspondence between invasive and non-invasive reconstructions are at sites that are very in close proximity to the actual ground truth implantation site. For the hippocampus, others did similar work using simulations or trying to localize epileptic spikes using MEG. However, in our work, we would like to do the same thing, only using mobile 64 EEG in the paradigm I described in the first part of my talk. So if you look at 64 channels EEG of one particular trial, you already can guess what we observe here. This is the data filtered in the theta range. And if you compare that directly to simultaneously recorded intracranial EEG, you already can see some similarities, but actually we want to quantify those similarities. So we do the same methods that I described before. We apply source modeling imaging, EEG source modeling, sorry, and then compute which sources actually corresponding best to the intracranial data. And if we do that, so if you compare to the left implanted electrodes, we can see a very smooth topography lateralized to the left. Here, positive voltages are red and negative voltages are blue. The nose would be on top. And if you look at the source modeling, what I'm showing here in yellow and red is reconstructed, non-invasively reconstructed sources corresponding to this intracranial data. And you already can see how nicely these localizations do match the implantation site as indicated as in these blue squares over here. So this was quite surprising to me that this even is working in mobile EEG. So if you change then the comparison from the left contact to the right contact, we see activities on the right. So again, this could be anywhere in the brain, but we only see sources that are correlating with the actual ground truth activities at close proximities to the implantation site. And that we did not only see in one participant, but also in another one. Again, we see very smooth topographies, localizing to areas that are very close to the implantation sites in the medial temporal lobe. And we actually analyzed 4 out of 5 participants where we have complete data sets so far. Looking at the left MTL, we see a left lateralization for all of those 4 participants. And then if we switch to the electrode in the right hemisphere, you already can see that the lateralization is beautifully changing, which is some sort of validation that we find the correct sources that we actually recorded in the intracranial data. So this is the localization. What about the functional data? You might remember these activities that happens before turns. So this turn approach theta activities that increase in all of our participants, we observed intracranially. So then we try to do the same thing non-invasively and compute the sources of medial temporal lobe activities, which I'm showing here below, and you can see how nicely these activities correspond between the intracranial and the non-invasive reconstructed data, which I do believe is quite promising to enlarge the participants included in our studies. So we might be combining intracranial and non-invasive methods to study a larger population of people. So with that, I would like to sum up that theta oscillations actually encoded the spatial geometry. Similarly, during real-world in imagined navigation, these oscillations seem to be internally generated because during imagined navigation there were no physical turns, environmental constraints, or visual flow. So these signals are internally generated and might support imaginations and episodic memory recollection at minimal sensory input. Scalp EEG actually contains activity of deep sources, which also then can be reconstructed using sophisticated source imaging techniques. And by using combined intracranial and scalp EEG, this enables novel perspectives to study cortical and deep brain activities in mobile setups. And with that, I would like to thank my lab that is led by Nanthi Suthana, which is an amazing supervisor, my whole lab team, which helped a lot during the recordings, all the funding sources, If you're interested in this work, the first part of the study is already published in this preprint I show over here. And if you're interested also, please contact me via email. And I would like to thank you for your attention. Thank you.